All right, hello everyone. Good morning, good afternoon, and good evening, wherever you are. Welcome to the Long-Term Animal Research Seminar Series. We continue this set of seminars with talks from Megan Petersdorf and Dr. Zarin Machanda. And before I introduce our speakers, I will make a couple of announcements. First, if you are in the US and you haven't voted yet, please make sure you do so. Secondly, if you're participating via Zoom, you should see a Q&A tab on the top or the bottom of your screen. If you open that tab, you'll be able to type any questions that you have, as well as see or upvote other people's questions. At the end of the talk, we'll go through those questions, starting with the ones with the most votes. However, if you have a clarifying question that you feel like needs to be addressed during the talk in order for you to understand something, you can type clarification in capital letters at the start of your question, and our speakers have said they will do their best to answer those clarification questions in real time. Lastly, recordings of all the talks will be available on YouTube shortly after they conclude, so if you need to leave early or know others who are unable to attend live, this talk will be available for viewing and reviewing after it's complete. All right, now that those logistics are done, I'm excited to introduce our first speaker, Megan Petersdorf, a PhD candidate in the Department of Anthropology at New York University. Megan studies the evolution of reproductive strategies and sexual signals in primates. She addresses these questions by combining behavioral observations in the field with endocrine, genetic, and morphological data. Her dissertation work focuses on female sexual signals in the Kinda baboon, a species which has received relatively little attention. So I'm excited to hear more about her work today. With that, Megan, I will turn it over to you.
Thanks, Liz. Thanks, everybody, for having me. I'm just going to share my screen. Cool. Great. I'm really excited to be here. I've been watching these talks all summer, and I'm just really excited. So thank you for having me, and Zarin, and thank you, everybody, for coming. So I'm Megan Petersdorf, and I'm going to be talking a little bit about my dissertation on the Kinda baboon at New York University. So before I begin, I just want to thank my collaborators, my supervisor, James Hyam, and all of my funding agencies, and just give a plug that you can find me and my lab on Twitter. Okay, so sexual selection leads to the evolution of traits that increase an individual's reproductive success and takes two main forms. Intrasexual selection leads to the evolution of traits that assist in competition among members of the same sex, and intersexual selection leads to the evolution of traits that are used in mate choice between the sexes. Most work is focused on one mechanism or one trait at a time, but less is known how selection mechanisms can interact to create the overall direction that sexual selection drives evolution. But multiple mechanisms can interact by reinforcing or opposing each other. So understanding the entire selective dynamic is important to understanding the evolution of any given trait or mating system. The starting point for this in large terrestrial mammals is ecology, which determines the distribution of females in time and space. Female reproductive synchrony is a product of group size and seasonality, and so it influences the extent to which a single male is able to monopolize reproduction among females in the group. We can think of reproductive synchrony occurring on a spectrum with different degrees of synchrony possible. But when we look at the extreme ends of the spectrum, some interesting patterns start to emerge. When there's a low degree of reproductive synchrony, a single male is able to monopolize reproduction, and this leads to high reproductive skew. In these systems, we see higher degrees of male-male contest competition selected for, which is reflected in large degrees of sexual dimorphism in body and canine size, and in their behavior, where we see competition over dominance rank. In these systems, we also see more indirect forms of female mate choice, where females' reproductive strategies often reinforce those of males. On the other side of the spectrum, when females have a higher degree of reproductive synchrony, it's more difficult for a single male to monopolize reproduction. This results in lower degrees of male reproductive skew. In these systems, we see weaker selection on contest competition and stronger selection for male scramble and sperm competition. This is reflected in lower degrees of sexual dimorphism, larger relative testis size, and in behavior where males acquire dominance rank through group tenure length rather than competition. So this leads to the highest ranking male being the male who's been in the group the longest, rather than the most competitive male. And finally, we see more direct forms of female mate choice where female strategies can often oppose the strategies of males. In large terrestrial mammals, we commonly see systems characterized by stronger degrees of contest competition among males. But in order to fully understand the extent of selection on phenotypic variation, we need more studies of large terrestrial mammals from the other side of the spectrum. So primates are a good model for this. And while they live in a variety of social and mating systems, reproductive strategies often vary in not only their presence, but the degree of expression, even in closely related taxa that live in the same social and mating systems. So this suggests that they've been subject to different combinations of selective mechanisms that have produced these really diverse phenotypes. So baboons in particular are a really good genus to test these types of ideas in. And there are 6 species of baboons that are widely spread across Africa. They can and do interbreed, but they show really interesting species-specific variation in their social and mating systems. So if we look at the baboons that live in multi-male, multi-female social systems, such as olive yellow and chocolate baboons, they appear to be under stronger selection for male-male contest competition. These baboons are generally characterized by non-seasonal breeding, high levels of sexual dimorphism, males who contest dominance rank upon immigration, and high male reproductive skew in favor of the alpha male. Kinda baboons are comparably less well-known baboon species that's found in Central Africa. And like chacma, yellow, and olive, they live in multi-male, multi-female groups with polygynandrous mating, but the more we learn about them, a slightly different picture starts to emerge. So their morphology is distinctive. They're the smallest baboon, and they have the lowest degrees of body size sexual dimorphism in comparison to the other species. Similarly, male canine size is also lower than in other baboons, which is driven by a smaller than expected canine size in males, which you can see in the plot in the bottom right, and you can see an example of this in the picture in the top left. They also have relatively larger testes volumes, suggesting an increased importance of sperm competition. They also interestingly live in really large groups, which range in size from around 70 to up to 300. And in comparison, other multi-male, multi-female baboon species often live in groups that range somewhere between 200 and 100 individuals. And lastly, they live in a really seasonal environment with a mean annual rainfall of around 1,100 millimeters. So based on kinda male morphology, behavior, and large group sizes in a seasonal environment, I set out to test the hypothesis that compared to other baboons, kindas have evolved under weaker contest and stronger indirect male-male competition, and kind of are occurring on this other end of the spectrum. So this leads to a series of predictions. First being that kinda females will exhibit a higher degree of reproductive synchrony in births and mating. Males might not compete directly over dominance rank, and instead rank will reflect group tenure length, and male reproductive skew will be shallow or unrelated to dominance rank. So kind of in sum, I'm predicting that they have increased female synchrony, which will lead to males not being able to monopolize reproduction, ultimately resulting in lower male reproductive skew. So I asked this question in a wild population of kinda baboons in Zambia at the Kasanka Baboon Project, which is currently the only field site on this species. The Kasanka Baboon Project was started in 2010 by Anna Weyer and has recently expanded to include multiple co-directors and collaborators, including myself, Kenny Chu, Jason Kamilar, India Schneider-Kreis, and Benjamin Mubemba. And also, none of our research would be possible without our team of Zambians on the ground, both past and present. They've taught us so much about the natural history of these really fascinating baboons, and we really wouldn't know everything we do without them. So to address these questions, we combined 8 years of long-term demographic data on births and dominance with 1 year of more intensive daily monitoring on female reproduction. So during this 1 year, we focused on cycling females and recorded all instances of mating. I also collected almost daily fecal samples so that I could later measure female reproductive hormones and determine ovulation and fertile periods for females across the year. So what did we find? We found that when looking at 8 years of birth data, births inversely pattern onto the seasonality and rainfall. So in this graph, the gray is rainfall and the green are the total number of births per month. So we found that 84% of births occur in the 6-month period between April and September. And this is in contrast to other baboon species, which typically show weak to no seasonality among reproduction. We confirm this pattern with circular statistics and the mean vector length, R. So when R equals 1, all births are on the same day of the year. And when R equals 0, all births are evenly spread among the year. And the mean vector length for births in our population was about 0.5. And for comparison, the Embacelli yellow baboons show a weekly seasonal pattern in their births, but their R value is 0.15. Now, if we pattern on the total number of cycles that pairs females have exhibited per month, we predictably see this opposite pattern emerging where mating becomes more common as we enter the rainy season. When we zoom in and look at the short-term data, we can get a better view of actual reproductive synchrony as measured on a daily basis. So when we're looking at the mating behavior data across the year, about 50% of observation days, there were 2 or more females observed mating, which is a commonly used metric in comparative analyses of synchrony. And for comparison, yellow baboon mating from the Embacelli show about a 25% overlap in female mating behavior. What's interesting is the way this distribution looks each month across the year. So while on average it's about 0.5, we see a slightly different pattern that's leading to this annual value. So I've looked at this in 2 different ways. The top graph is the distribution of sexual swellings, and the bottom graph is the distribution of mating, just so you can kind of have a different visualization. On top, the gray bars are nulliparous females and the black bars are parous females. And on the bottom, the light green is nulliparous plus parous females, and the green is just the parous females. So the bottom is this same metric I was talking about before, the proportion of days in the month where more than 2 females were observed mating. So these are monthly values plotted that were calculated from daily data. So you can see that in some months, the values are 0 or close to 0, but in other months, they're close to 1, which is leading to this kind of on average value of 0.5 annually. And you can also start to notice this pattern that mostly the parous females are exhibiting this pattern. Nulliparous females are often beginning their cycling outside of this peak mating season. So moving on to male dominance rank, their dominance system is also really unique. So we used ELR ratings over time to determine dominance rank. And across our 8 years of long-term data, we found that the alpha male tenure length is really long, often averaging multiple years. In other baboon species, you see a higher turnover rate in the alpha position, given that it's contested. So we also found that immigrant males enter the dominance hierarchy at the lowest rank and rise in rank over time, which is something we don't see in other baboons. In other baboons, when males immigrate into the group, they try to join a group where they are able to contest the alpha male for that position. And actually, these data are from 2008 or 2018 when I was working on this paper, but our current alpha male is still the alpha male, so he's been that position for almost 5 years. Finally, we looked at how mating success was distributed among adult males in the group. So we first looked at the B index, which determines if mating is skewed towards a single male in the group, regardless of their dominance rank. And this index varies between -1 and 1, with values closer to 1 reflecting high skew, -1 reflecting an equal distribution, and 0 is a random distribution of mating among the males. So our B index was pretty close to 0, which means our distribution is not significantly different from random. And when you do more tests to compare minimum and maximum B values from simulations to your confidence interval, we can also reject that the distribution is skewed, but we can't reject that the distribution may also be equal. So male mating is either random or equal, but it's not skewed. So what does this look like when we do consider dominance rank? So first, when we plot the predicted skew according to the priority of access model, which takes into account the inherent female synchrony and allocates reproductive success in order of dominance, we do see a shallow relationship, but the alpha male is still predicted to have the highest success with about 50% of mating and parenting or paternity opportunities. But when we look at actual mating success, we see that it's pretty evenly distributed other than the lowest ranked male. Which is interesting, with the alpha male not getting the highest and getting about 25%. So in other baboons, And in Canine baboons, multiple males are often able to mate outside of the fertile phase, but the alpha male is able to monopolize mating in this critical time. So we also restricted our data set to this 5-day fertile phase during the cycle, which we were able to determine from hormonal data. And when we do this, the same pattern emerges, with there being no correlation with dominance rank, and the alpha male still on average getting only about 25% of mating opportunities across the females. Okay, so in summary, all of our predictions were supported in favor of our hypothesis. So large group sizes and seasonal environments are leading to synchrony in female births and mating. Males aren't competing over dominance. Dominance is reflecting group tenure length, and male mating success is not skewed and does not correlate with dominance rank. The next steps include not only looking at male mating skew, but also reproductive skew and paternity. Perhaps we'll find a different or similar pattern when we look at the paternity of the infants. Unfortunately, we're able to look at this annually for 7 years of genetic data to get a better idea of the skew patterns. And I'm really excited about this, and we're really close to finally getting these data. We've had some kind of bioinformatics snafus, but I know that we're close. So hopefully I'll have that information soon, and we'll be able to not only look at short term how mating success translates into paternity success or not, but also long term if these patterns are showing up the same every year. So in summary, it's important to understand how selective mechanisms interact to drive evolution in different ways by looking at the entire dynamic of a mating system rather than focusing on one trait or one mechanism at a time. My dissertation is kind of taking this more holistic view beyond just these kind of long-term patterns. But this can give us insight into these feedback loops that might be occurring among male and female strategies and how they interact and have co-evolved, and ultimately produce this extraordinary variation in phenotypes that we see. So importantly, the study shows that differences in the environment can select for divergent phenotypes, even in closely related species that live in the same social and mating systems, which shows us again that sexual selection can be a major driver of phenotypic variation and diversity. Thank you so much.
Thank you so much, Megan. I think we'll start off with a question from Matthew.
Yeah, I have at least 3, but I'll just start with one. So you showed that data for the nulliparous versus multiparous females and their seasonality, and the nulliparous females were not especially seasonal. Is that true if you restrict it to just their conceptive cycles? Are those seasonal?
Yeah, their conceptive cycles are a lot tighter as well.
So why do you think that is? I guess is my follow-up. What's happening in those first few cycles, or however many that aren't in the right season?
Yeah, that's a good question. And I think a lot of people exclude nulliparous females for this reason, because they don't fit the patterns that we expect. And one of my undergrads is actually doing her thesis on this kind of differences. So I don't have a ton of answers for you right now, but I definitely think it's something about, we know that nulliparous females have to kind of ramp up their reproductive cycles, and they often exhibit more cycles. So I do find it interesting that they kind of conceive around the same time, and so they're ready, but they have to start earlier. And what's interesting is when I looked at seasonality metrics for resumption of cycling in Paris females and seasonality metrics for like the onset of menarche, you don't see seasonal patterns in the onset of menarche at all, but you do see seasonal patterns in the Paris females resumption of cycling.
Cool, thanks.
All right, so we have a question from Susan Alberts, who says, first, great talk. Maybe you mentioned this, but I wonder how you think about Canine baboons compared to some of the macaque species, such as rhesus macaques, or generally other seasonal terrestrial circumpothecines. Do you think these kindas are like macaques in social behaviors?
Yeah, that's a great question. So a lot of these ideas were built by my advisor, James Heim, and his former postdoc, Constance Dubuc, and they built them around a lot of the research on macaques. And I think people who study macaques have been kind of taking these approaches for a while. And I definitely think that they're a lot like macaques in these ways. So I would say that the Canine baboons are more similar to rhesus macaques, whereas something like a chacma baboon is similar to a crested macaque.
All right, great, thank you. And I think with that, we'll switch gears. Thank you for giving such a great talk, Megan.
Thank you so much.
All right, so for our second talk, I'm very pleased to introduce Dr. Zarin Machanda. Zarin is an assistant professor at the Department of Anthropology at Tufts University. Her research focuses on the factors that affect the development and quality of social relationships in chimpanzees. She has worked on the evolution of male-female relationships, male-male cooperation in hunting, and how chimpanzees use social communication to mediate social relationships. What really impresses me in particular about Zarin's work is how she combines different metrics of sociality to more deeply understand social relationships. So I'm thrilled to welcome her today to hear more about chimpanzee social behavior. Take it away. Oh, you're on mute.
Now, can you hear me?
Yes.
Okay, great. Thank you. First of all, hopefully you can see my slides. And I just want to say first, thank you to Liz and Matthew for the invitation. I'm really excited to be here today. And what I'd like to do with the talk today is actually share with you some of the highlights from research that we've conducted at the Kibale Chimpanzee Project over the last many decades, and show how we can use some of these pretty incredible data sets from long-term field studies of primates to answer different questions about behavioral ecology, but also to expand our knowledge into even understanding different fields like human psychology. So I just wanted to start with a little bit of information about the project. So the Kibale Chimpanzee Project is located in southwestern Uganda, and we study the Kanyawara chimpanzee community located in Kibale National Park. The research project was started in 1987 by my PhD advisor, Richard Wrangham, and we've been systematically collecting data from these chimpanzees since 87. And today the project is directed by Richard, myself, and collaborators Melissa Emery Thompson and Martin Muller, who are at the University of New Mexico. We have a pretty extensive set of data collection protocols at the Kibale Chimp Project, including full day focal follow. So we follow individuals from the moment they get up to the moment they go to sleep every single day of the year for about 12 hours a day. And we take scans every single minute of the day. So this is a pretty detailed data set of the daily lives of these 50 to 60 chimps. We also have kind of long-term group records from 1987 that really haven't changed that much since 1987. Basically what happens is a team of 2 field assistants goes out every day in the forest. One of them chooses a focal to follow, and the other takes detailed records of everything else that's happening in the group. Along with that, we have detailed records since 1996 of health and physiology, including a biobank of somewhere between 40 and 50,000 urine samples that are at the University of New Mexico that we can get a lot of detailed physiological data from. Now, this data is all in a relational database that I store and maintain at Tufts. And we're a little bit old-fashioned, I would say. We actually collect our data still using pen and paper in the field. And so you don't really need to know exactly what's happening on these check sheets, but here are just some paper check sheets that kind of illustrate the focal data. So here on the left, we have an adult focal follow. On the right, we have an infant focal follow. We take data not only every minute on what the focal is doing, but also all aggression, population, all play behavior, any toy play. So pretty detailed records of the focal, pen and paper. Here are what the behavior follows look like. So it's basically kind of fun because it's just like handwritten notes of everything that's happening in the party that we're following at the time. So just to give you some highlights here from this day in 2014. This is a morning. We have Johnny charging and chasing and attacking Yogi Bear with kicks and slaps, and Yogi flees screaming. We have some health data. So every cough, every sneeze is recorded. We have every copulation. So here's a copulation with screams and darts from Lonjo with Rwanda that lasts 8 seconds, which is a whopping 1 second longer than our average copulation. In this case, the female approached the male. We have data on some intergroup interactions where the chimp. And in total, with all of this data from 1987, have just over 95,000 hours of behavioral data on 150 individuals. And actually 95,000 hours goes till somewhere between, somewhere in like the middle of 2018. So we're still waiting for some of our 2019 data. So we're hopefully over the 100,000 hour mark with our data set, which is very exciting. As I said, we have this biobank of somewhere between 40 and 50,000 urine samples where we're able to assay all sorts of things, testosterone, estrogen, progesterone, human chorionic gonadotropin, which is kind of a pregnancy hormone. So basically, actually, we can do this in the field where we take human pregnancy tests and dip it in the female chimp urine. Chimps and humans are so closely related that we can actually figure out if our females are pregnant with human pregnancy tests. In the lab, we can do cortisol, C-peptide of insulin, which is a measure of energy balance, creatinine, which is a measure of muscle mass. And then more recently, we have added assays to look at things like thyroid hormone and oxidative stress. We also have some interesting new photographic field methods to track things like morphological development, including using laser photogrammetry to get measures of body size and growth. And one of my favorite projects, which is a project that will help us better understand dental development and kind of tooth wear, which involves over 9,000 photographs of baby chimps with their mouths open to figure out when they are getting their teeth. So that was a fun and hard data set to collect. I can tell you a little bit more about that later. One of the things that I wanted to highlight with my talk is the fact that we can use long-term data from these wild populations to really understand development and the study of life course. So really understanding how the life course changes with age. So we have data from infants who were born into our field site who are now adults. And this long-term data allows us to ask some pretty detailed questions. And I'd like to highlight Kanyawara and KCP as one of the populations, one of the primate field sites where we have this data on a wild population of chimps with decades of long-term data and kind of multiple data sets that don't just look at behavior, but where we can integrate behavior with other measures. And that's what we have with Kibale Chimpanzee Project. So I want to kind of show you some examples of how we can use these multiple cool data sets to get at particular questions of life course change. And I'm going to focus on two time periods, really early development and then old age. So anytime we kind of think about how development occurs or how lives change over the course of time, we're really rooting these studies in life history, which is about the allocation of resources. So you have a set kind of amount of resources in a habitat, and individuals have to use those resources and really divide them up with among these three processes of growth, maintenance, and so in early development, we really kind of focus on growing and investing a lot of resources to growth. But then as we mature and enter adolescence and adulthood, we shift resources away from growth and into reproduction. For humans, especially as we age, we then start to shift resources away from reproduction and then really focus on maintenance. And so that's something that's more likely to happen in old age. I would like to argue that the chimpanzee data are really essential for understanding aspects of the evolution of human life history. So human life history is notable for its incredibly long periods of development, early development, so a long juvenile period in particular, as well as this long period of post-reproduction. Now, chimpanzees in the wild can live between 50 and 60 years. So we do have these opportunities with long-term chimpanzee data to really understand not only early development, but also this kind of process of aging. And chimpanzees kind of reach their peak what we call prime age around the age of 30 to 35, but then they might have, up to 2 decades of life after that, where they are kind of living in this post-prime time. So, I think chimpanzees are an interesting model to really understand human evolution and the evolution of human life history. So just a couple of details about chimpanzee social behavior. One thing for people who might not know, adult males are more social than adult females. This is possibly a little bit different from some other species, but it's not uncommon for us to see a lot of very kind of affiliative behaviors amongst our adult males. So here we have a video of Makoklu and I think that's Kakama, who's the alpha male on the left, grooming each other. And we're very likely to see adult males groom each other during the day. So I've done a couple of studies where we've looked at, as Liz has said, about different kinds of metrics of social behavior. Here's a study that we did just comparing the strength of social bonds among adult males and adult females. And so we can see using different metrics on the left of party association, so time spent in subgroups. In the middle, here is data on time spent in proximity to one another, and on the right is data on grooming. And for all of these metrics, what we see is that male-male dyads show stronger metrics of affiliation compared to our female-female dyads. And actually doing these studies was really interesting because, especially at Kanyawara, our females really don't groom a lot. So in one of our studies, we had something like 2,000 grooming bouts, 2,048 grooming bouts among adult individuals. And of those, only 32 were between adult females. So they are actually, at least at Kanyawara in terms of grooming, pretty asocial compared to our males. Now, I wanted to kind of think a little bit about early development and the development of this kind of sex difference in sociality. And so we wanted to think a little bit more about when males become more social. So is this something that we see early in development or later? And there are kind of two hypotheses we can think about here. One is that we might actually start to see the sex difference in sociality as adults, because as youngsters, females transfer into new groups at adolescence. So before that point that they transfer, immature males and females are actually both in groups with kin. So you might expect that there would be no difference in sociality because everybody has the opportunity to interact with kin. The second hypothesis is that actually you might see these changes in sociality during the immature or subadult period, because what chimpanzees are doing is using the subadult period to practice future social skills. And since males do use or engage in more sociality as adults, you might see kind of more sociality developing through this immature period. And so here's kind of an example of how we can use this long-term data to really understand some of these patterns. So I'm just showing you actually data on association patterns. So this is time spent in a party with their individual's mother in purple versus time spent in a party with adult males and not the mother. So in many ways, this is kind of a measure of independence. And here is data just for one of our individuals for whom we have the entire kind of span of the immature period. This is Kalimi's data. Kalimi transferred into a new group around the age of 11 and a half. And what we see when we look at Kalimi's pattern is that she is really a mama's girl. She spends almost 99% of her time in a party with her mother. There's really very, very few observations of her with adult males without her mother present. Now, so this is kind of an example of a female pattern. Let's look at Lonjo, who, again, we have data from Lonjo from the time he was born, actually from the day he was born into his adult period. And here we have a very different pattern. So what we're finding is that, between at around the age of 9, Lonjo starts moving away from his mother into parties with adult males without his mother present. And really, between this age of 9 and 12 for Lonjo, we see this shift towards more sociality with adult males or more independence. But what we are able to do is actually, because we have long-term data, we can look and average these patterns across multiple individuals. And so we can show here the data for 32 females from our long-term data set. And essentially, we're showing something very similar to Kallimi. There are some females who are going to be a little bit more independent than Kallimi, but essentially, these females are spending a lot of time with their mothers until they transfer to new groups. For males, on the other hand, you see kind of the same pattern that we saw with Lonjo, where, you know, starting at about the age of 8.5 until about the age of 12, we see this shift towards more independence, more time spent with their mother. And so what we're finding is males do kind of become more social during this period of late juvenility and adolescence. So males are showing more social behavior than females, or at least more time, more independent time with adult males. And possibly this is a way for them to start forming social bonds with adult males and making their way into the dominance hierarchy. But what I'd like to argue is that we can actually use a more integrative approach with these long-term data sets and understand multiple aspects of development and how they interact with each other, not just the social data. So for example, we can use our photographic data set to look at something like canine development. And so here's a nice picture of Bono with his mouth nice wide open. I would like to stress how hard it is to actually get clear pictures of chimps, especially baby chimps with their mouths open. So it was a very cool project to work on. It's kind of also just a nice science story. This is one of those projects that came about because we were very interested in when chimpanzees get their different teeth, how that compares to humans. This is partly because the human fossil record has so many teeth in it. And usually the traditional way of doing this is to wait for baby chimps or immature chimps to die and then collect their skeleton. And we were actually talking with someone who's just like not a biological anthropologist about, oh, how cool would it be to get dental development data on these chimpanzees? And someone said, oh, why don't you just take photos of baby chimps with their mouths open? And we were like, why don't we take photos of baby chimps with their mouths open? That's amazing. And so 9,000 photos later, we have some really cool data on dental development in these chimps. But in particular, with here is an adolescent male, what we were able to find out with our data set. So we had 3 adolescent males who erupted their canine teeth between the ages of 10 and 11. And these are canine teeth are in many ways fighting teeth. So just keep some of these numbers in mind. As I already said, we actually also are able to collect data on growth and body size using parallel laser photogrammetry. This was actually a study that we got one of our undergraduates involved with. And again, I'd like to also highlight one of the great things about long-term data sets and these long-term field projects is our ability to bring in all sorts of people to work on this science. So from undergraduates, also high school students, we can actually get a lot of people involved in this kind of work. So parallel laser photogrammetry is a really interesting and cool tool to assess non-invasively body size in wild animals. It was first developed, as far as I am aware, with seals, who are a little bit more cooperative than chimpanzees. But what we do is have this parallel laser system that sits on top of a camera. These lasers are always, in our case, 4.7 centimeters apart. We can shine them on the animals, take pictures of them in appropriate positions, and then we have a ruler in every photo. And so we're able to assess things like body or trunk length, which is a metric, for example, that's a proxy for skeletal length. We do this multiple times a month for all of our individuals, and so we're able to compile a pretty robust data set on body size. This has allowed us to model things like growth curves, and so I'm not going to show all of the details here, but here's an example of a trunk velocity curve that we're able to put together. This is for trunk area. And what we were able to show here is that there is a growth spurt in chimpanzee muscle mass for the males, not really for the females, and that we see this growth spurt or this peak in the velocity curve at around the age of 9 years. So that's also kind of interesting. So again, keep some of these numbers in mind. From our biobank, from our physiological data on testosterone, we are able to show that there's an increase in testosterone for males between the ages of 9 and 17. And then again, some more behavioral data, just looking at when aggression increases. What we're able to show is that aggression, especially the sex difference in aggression, is starting to come about around the age of adolescence. So we don't see a sex difference in aggression, a very strong signal of this before the age of 10 in this particular data set. It's coming out a little bit more strongly in adolescence, and then you can see the adult data here. So there's almost no aggression from this age of 0 to 10, but then we start to see aggression increase, especially for the males in adolescence. And I would just like to point out, this is a very characteristic sex difference across a number of mammals, especially chimpanzees, where we have a sex difference among the adults in aggression. So adult males in this particular year that I'm showing are about 11 times more aggressive than the adult females. And in some years, it's actually up to 14 times more aggressive. So what we're seeing is that development of aggression, of sex differences and aggression starting around the age of adolescence. I'm going to actually skip this just to keep going. But I wanted to kind of take all of this together. And what we're finding is, we know that males begin in integration into the, somewhere starting around the age of 9. That's when we start to see these males becoming more independent and moving away from their mom. That's also around the time that we see an increase in aggression. And we also know that we're seeing differences, or at least that's when they get their canine teeth. That's about the time that they have this growth spurt in muscle mass. And this also corresponds to an increase in testosterone. So I think what's kind of interesting is that we are seeing not just changes in behavior, but that we can use these long-term data sets to integrate different types of data to tell a more complete story about development, and also understand some of the underlying mechanisms that might be playing a role in some of these behavioral changes. So that's kind of a nice story about how to look at early development with long-term data sets. I want to kind of shift a little bit and now think about what's happening at the end of life for these chimpanzees. And I want to focus a little bit more on social bonds. And we know that chimpanzees have these preferred social partnerships. And so these are for males. We know that male chimpanzees have some very strong mutual bonds that they develop in adulthood and through to the end of their lives. And we know that these, what we might call friendships, we sometimes refer to them as preferred social partners, have lots of kind of interesting correlations with the rest of our data. So mutual friends, preferred partners tend to groom more equitably. They do more joint displays, form more coalitions. We all have some really interesting data with vocalizations where they're more likely to chorus with one another, and actually they're more likely to change the sound of their vocalization to match their partner. And a fun study at a different field site, we were actually able to show that these preferred partners were more likely to actually warn each other of danger when faced with a model snake, so that they were more likely to give predator alarm calls to their friends compared to their non-friends. So this shows that chimpanzees can be big jerks, but also that these kind of social bonds can pattern a lot of their other behaviors. We also know, and this is not just true of chimpanzees, but lots of other species, including baboons, that social bonds are important to reproductive success. For male chimpanzees, we know that social bonds aid with mate guarding, formations of alliances, gaining rank, maintaining rank, and that social bonds can kind of pattern reproductive success in terms of concessions by the alpha male to their to their social partners for in trading basically copulations. So we wanted to kind of understand, we do a lot of work on trying to understand how and why these males are forming social bonds. And because we're interested in aging, we wanted to ask questions about social aging and how these social bonds change with age. And so actually what we did was on social selectivity in aging in wild chimpanzees, and this actually just came out in Science about a week and a half ago. So if you'd like to read the paper, it's just recently come out. This is actually work that I think also is kind of a fun science story for those of us who do long-term work on long-term field sites. So this research was led by me and Dr. Alex Rosati, who's a professor at the University of Michigan. She actually is an alum of Duke. She did her PhD in evolutionary anthropology. And it's kind of a fun study. So Alex and I have been friends since 2004. So actually, as we've aged, our social bonds have grown stronger, which is kind of fun. But also, Alex is a psychologist who does a lot of work in animal cognition. And we would go for coffee because we're friends and we'd talk science. And she and I were talking one day and she was asking questions about our long-term data. She said, I think your long-term data would actually be really interesting to test this theory from human psychology called socio-emotional selectivity theory. Now, as a behavioral ecologist, I had never heard of this theory. I didn't realize how interesting or important it was. And so I think what's kind of fun is that what I'm going to show you is an example of how long-term field sites can be used, and the data from these sites can be used to answer questions that are outside of behavioral ecology, and that they can actually impact pretty diverse fields. So socio-emotional selectivity theory is a theory that has been developed from human psychology to explain shifts in social behavior and cognition during aging for humans. Now, there is a characteristic social aging pattern or phenotype in humans, which is that as humans age, they show a positivity bias. So this kind of stereotype of grumpy old people is actually pretty incorrect. And across cultures, humans actually get more positive with age. Humans also become more selective with age in that they basically kind of focus on existing relationships rather than developing new relationships. And so they kind of have smaller social circles, but they report greater investment and emotional satisfaction from the bonds that they have. Now, this has kind of been explained in many ways that as humans age, when they're young, you need to focus on increasing your knowledge. So you have lots and lots of social bonds, even though some of them might be negative because you're learning things. But as you age, you kind of know all you need to know, and you focus more on your emotional needs and just kind of think about fulfilling bonds and being very positive. So we see these characteristic shifts in humans. Now, the explanation for this, so socio-emotional selectivity theory, is basically what they do is hypothesize that these shifts in social behavior in humans are grounded in our ability to monitor time, have future time horizons, and have this knowledge of mortality. So as humans age, this is my father who, is showing. Probably actually he's very grumpy in this picture, but he moved from India to Canada in 1967 and has been cold ever since. So I think he was just grumpy. He's just been grumpy because of that. But basically the idea is that as humans age, they are, they can project themselves into the future and see that time is running out. And they don't want to waste the time that they have by focusing on negative things, negative stimuli, or by investing in negative relationships. So socio-emotional selectivity theory basically says that because humans have this ability to monitor time, and this knowledge of mortality, that's why we see these shifts. So what Alex and I thought was, well, wouldn't it be interesting to look at chimpanzee data? Because there's no evidence that chimpanzees have this ability to monitor time or have this sense of future self. So they are maybe able to project themselves or plan for future things, maybe a few hours or a day in advance, but there's no data that suggests that they can kind of think of themselves in the future on the scale that humans do it, which is, over years or decades. So we thought this would be interesting because if chimps and humans show the same pattern, we would have to reassess the key assumption of socio-emotional selectivity theory. But if chimps and humans show different patterns, this actually then provides comparative support for the theory. So we thought this is kind of an interesting way of looking at long-term data from a wild population of animals to actually say something about human psychology. So for this project, we used data for 20 years on 21 male chimpanzees. We focused on males here because they do form those strong social bonds, and it allowed us to use quite a diverse series of metrics for this study. We first wanted to just ask the question about looking at what are the general patterns of social aging, be the same positivity bias that we see in humans. And what we found is that grooming, which we kind of consider a positive or affiliative behavior, increases age, but it more or less stays fairly consistent. But importantly, what we found is that aggression, which is a negative or agonistic behavior, decreases with age. So whether we look at directed aggression, which has a target, or non-directed aggression, which are just things like dominance displays, we see declines in this with age. And so taking these things together, what we are finding is a shift towards more positive behaviors with these wild chimps compared to negative behaviors. So that is something that mirrors what we see in humans. We also wanted to ask if social relationships shift with age as well. And we did this using a number of metrics, but for the sake of time, I'm not going to go through every single one with you today, but specifically, we were very interested in these friendships that were formed, and we were able to calculate different kinds of friendships. The first were mutual friends, and we used a metric of proximity to define friendship, so individuals who are sitting more closely together. And what we did was we looked at individuals who kind of reciprocate this proximity. So who are more likely to kind of sit near each other when each one was the focal compared to their own average of sitting next to anybody. And we also looked at lopsided or unidirectional friendships. And the way I think about unidirectional friendships are like kind of thinking about young siblings and older siblings. So young siblings are always wanting to sit next to their older sibling, and the older siblings always moving away and wanting to sit next to one of their friends. And so you would say that this is a lopsided relationship because one individual is really motivated and the other does not really reciprocate that. And what we were actually able to find, what we found is that as individuals age, they increase the number of mutual friends that they have. And young individuals were more likely to have lopsided friends, and old individuals were more likely to have these mutual friends. And so that's kind of a very interesting difference. Everyone kind of has the same amount of friends, but the nature of those friendships shift. And what was also interesting is that everyone actually really likes being friends with the older individuals. So older individuals are really sought after as social partners. So this is a graph of mutual friendships, and you can see actually here that older adults tend to have mutual friends who are also older adults. So these mutual bonds tend to be between these old individuals. And then we also show that younger individuals are kind of really attracted to these old males. So unidirectional, when younger individuals form unidirectional friendships, they're often doing that with older, either prime-aged or older-aged adults. So this suggests that there is something kind of really interesting and potentially valuable about these older individuals. They have some kind of social capital that we still need to better understand. So finally, we just wanted to look at whether older individuals change their investment by looking at a grooming metric. And what we were able to find is that everybody, whether they're young or old, actually grooms their mutual friends more than either non-friends or lopsided friends. And just keep in mind that, and we did this with a number of grooming metrics, and just keep in mind that, remember, these older adults groom mutual friends more, and they have more mutual friends. So in many ways, they are kind of shifting their behaviors towards these more positive reciprocal relationships. I want to point out that this is happening even though these older males are losing dominance rank. It's also happening, this is data from our creatinine studies, this is happening during a time when males are also losing muscle mass, which we get from our creatinine data. So they are kind of losing body condition. Their testosterone is decreasing. And so we do see that, this is not surprising because we're seeing muscle mass and testosterone probably going hand in hand. But at the same time, their reproduction is not necessarily changing that much. So old males, even into their 50s, are still actually able to maintain copulations with both with Paris females who are the more attractive females. So there's something that these males are doing right. Like, they're actually living a pretty good life despite losing body condition and rank. They're maintaining these friendships and able to actually maintain some reproduction. So we are finding a positivity bias. We are seeing a shift in social relationships with age towards more reciprocal friendships, and they are investing in these mutual bonds quite a bit. And they're doing this despite losing rank. But they're, and so there is something kind of really interesting about these old males. And so this is actually one of the first times that this is the first study where we're showing in a non-human organism a similarity in social aging patterns to humans. So we're finding that humans and chimpanzees show the same pattern in the absence of mortality. And we still need to do a little bit work to understand why we might be seeing these same patterns if it's not being explained by this kind of shared future time horizon. So we're arguing that this might have to do with a shared sense of emotional regulation between chimps and humans. There is data on social aging in other primates, especially from baboons and macaques, although I would like to put out a call that we need to study aging in lots and lots of different species to better understand these patterns. There are really interesting age and dominance patterns that's pretty highly variable across other primates. Alex and I just finished a review paper that's out that looks at some of this. Most other primates where we were able to find data about aging actually show declines in sociality. And actually, most of the evidence right now shows a negativity bias, so that grooming actually often decreases with age, where aggression stays relatively the same. So monkeys tend to show a very different pattern from what we're seeing with humans and chimpanzees. And there's almost no evidence of kind of selectivity in aging primates. So there are a few studies that have shown changes in social behavior in other primates, but none that show the same kind of positivity bias or selectivity pattern that we're finding in the chimps. So now we need to look at lots and lots of other things. We'd like to look at how coalitionary aggression changes with age to see if that's how old males are maintaining their reproductive success. And of course, we really want to look at females to see if females are showing the same pattern. So I just want to finish by hopefully highlighting how valuable these long-term research sites on wild primates can be. We're not the only field site. We're not, obviously, Gombe has a lot of decades of data. The Amboseli baboons are kind of just this amazing field site with data for multiple generations that we all aspire to have. But I do think that one of the cool things about these long-term field sites, where we're able to collect data on the same individuals over many, many decades with multiple data sets, does help us understand some interesting questions about both early development, but also aging. And I do think that at least the aging study that I showed you is a really nice example of how long-term field sites or data from those field sites can be used beyond behavioral ecology to help us and to say something outside of our field, and in this case, in human psychology. So as Megan said, none of this work could be done, it would be at all possible without our Ugandan colleagues and the staff of the Kibali Chimpanzee Project. They collect this data. They have saved my life on several occasions. I'd like to also thank the other directors of Kibali Chimp Project, Richard Ranga, Martin Muller, and Melissa Emery Thompson, and of course our funders, most recently the National Institutes of Aging and NSF. So thank you very much. And you can also follow us on Twitter at Kibali Chimps.
Great, thank you so much. There's some questions coming in. First, I want to start with a question that both Matthew and I had about how you measure aging and social relationships. So if the increase in positivity as individuals grow older is due to maybe awareness of impending mortality or not, you might predict that the time to future death might be a better predictor of the nature of social relationships. So have you been able to compare the ability of like chronological age compared to time to death to predict changes in social relationships?
We haven't done that. I mean, I think that would be interesting to look at, so one of the other things that we've been working on are measures of frailty. So Melissa Emery Thompson and our team has a paper recently out this past summer on frailty. And actually what's kind of interesting is that we don't really see kind of frailty in these chimps. Like they're pretty incredibly robust. So you don't see these kind of long declines or they're looking really like scraggly and like doing poorly. These chimps are doing remarkably well. But I mean, I think it would be interesting. There are some chimps who you're kind of like looking at, you're like, oh, they're not doing super well. Maybe we should. So I think that would be an interesting way of approaching it. We did not look at that. We just looked at chronological aging. But yeah, that could be interesting.
Okay, great, thanks. Delphine Demore says, first, thank you for the nice talk. And could you please explain a bit why you chose to categorize relationships based on proximity data and not include grooming? I think that those males that are both initiating social proximity and groom more longer and more equitable might be actual friends.
Right, so partly why we couldn't do that is we wanted to use the friendship data to predict something about grooming behavior. So we actually had to have the friendships be independent of our grooming data. Otherwise, we would just be saying, do grooming relationships predict grooming relationships? So we needed to keep those independent. But I actually do think proximity is a good measure of friendships. I often think about it when I talk to students about it. I say, now imagine if we followed you around every minute of your day for like every year of your life. You probably would sit closer to your friends than your non-friends. And I think with chimpanzees in particular, sitting in proximity does put you into a vulnerable position because these are aggressive animals. And if an individual is going to be aggressive, you are the closest target to that individual. So I agree. I mean, I think friendship metrics in other studies, we would probably include a grooming variable. We'd probably do some kind of composite measure, which is pretty traditional in primate studies. But in this particular case, we needed it to be independent of grooming.
Great, thanks. And as a follow-up question, Delphine asks, do you find selectivity for mutual friendships is driven by the older males or just that older males have more mutual friends?
So I'm not totally sure I understand the question, but I think, so what we are finding is that these shifts are happening kind of throughout the life course. So like, I mean, hopefully you saw that it's kind of an increase, that this increase from the age of 15 into old age. But it's not, the result is not just because these young males are showing few mutual friends. So there is a difference between old males and prime age males, so it is not just being kind of driven by prime age and past prime males being different from the young males, but actually these old males doing something differently than the other adults. So we don't have, as part of our long-term data, any information on kind of approaches or retreats where we could look at, something traditional like a hind index, where you could look at who's really like maintaining these things, which is why we kind of have this other metric of kind of mutual versus lopsided. It'd be great to have that kind of approach in retreat data, but we don't have it. I'd also like to point out, I think that brings up one last thing that I'd like to say about long-term studies. We obviously didn't start Kibale Chimpanzee Project in 1987 to test socio-emotional selectivity theory. And if we had, we probably would collect different data. And I think one of the differences for all the PhD students who are at the seminar between, designing data collection for a PhD versus long-term data collection. With long-term data collection, we have to design data collection for questions we don't know we have yet, right? For questions that we're going to have in 10 years. So it has to be kind of broad, and there has to be a trade-off between breadth and depth. And, it does mean that sometimes when we want to test certain things, we don't have exactly the perfect data set that we'd want. We have to use what we have. But that's one of the trade-offs. I mean, 10 years from now, we're going to have a new question that we just don't know about yet, and we hopefully will be able to look at that with the data we have.
That is an excellent point. And I think just one last question to end today from Ann PC, who says, first, lovely talk. And then she wonders about old females. Do you have any thoughts about how their social relationships might change?
Yeah, I mean, so hi, Ann. It's nice that you're here. So our females are fairly asocial. Like I said, with the grooming data, it's pretty hard to get a sense. We do have a couple of females who have kind of one or two very strong relationships. They do tend to be some of our older, higher ranking females. I think what we have to do with females is consider not just bonds with other adult females, but how their social relationships across all sorts of individuals change. So we'd also look probably at how they behave with their offspring and how mother-offspring relationships change with age. And I think the reason for that is that the human data actually do not explicitly say anything about the type of relationship. So they're not just looking at non-kin bonds, for example. The human data kind of just shove all these bonds together, whether it's like your spouse or your relationships with kids. And so I think we would have to look at the chimp data, the female chimp data, with keeping that in mind, and also including things about how their relationships with their offspring change with age. And I don't have a gut feeling about that. I bet Anne does. So I'll probably have to email her and talk to her about that later. But it is true that I think we're going to have to use slightly different metrics to look at this female stuff. The very, very, very limited data that is out there about female chimps and aging suggests actually that they may show the opposite pattern to males. And there is some data from other species like lemurs with social aging where males and females do show opposite patterns. And so that's why we're particularly interested in this, in the females.
All right, thank you so much. So I'd like to thank again Megan and Zarin for providing us some much needed distraction today and talking about some really interesting results. And to everyone out there in the Zoom and YouTube universe, we'll see you next Tuesday, same time, same place for more interesting talks. Thanks all, take care.